Topic Notes 5.1 Classification and Embryology. So it's time for us to dive into the animal world. And before we get too far in, we need to understand how we name and classify things. For example, this is a picture of a tunicate, and it's actually our closest invertebrate relative. How do we know that? From a lot of really good research that goes into looking at the genetics and embryology and essentially the evolutionary connections between organisms. So let's get started. The main idea here is that the Linnaeus taxonomic system allows scientists to use a standardized system to identify a group of living organisms and to study evolutionary connections. And of course, always remember to pay attention to each of the individual learning goals. So why do we classify? Here's the thing. Not everything is what it looks like or seems like on the surface. For example, the picture to the left there are penguins. Now, we know they're birds, but they don't fly. Over on the right, you have a picture of a bottlenose dolphin. And of course, dolphins we know are mammals just like us, right? They breathe air, all that. But they have fins and they swim in the water. So they people even today call them fish. The kelp in the middle picture there, that's actually algae, yet it looks like a vascular plant like that we would normally see on land. To say it's algae seems very strange to us even now. But that's why classification is so important. It allows scientists to talk to each other about these various different organisms because, of course, we range from the microscopic to the largest animals on the planet. And it's just a huge amount of diversity to try and organize and understand and discuss and study. Not only that, it helps us to understand the evolutionary connections between all of these animals, and that's something we'll kind of touch on as we go through the different animal phyla. So here's the big definition. The taxonomic hierarchy, the classification of the species within living organisms by describing the domain, kingdom, phylum, class, order, family, genus, and species. Now, early efforts to describe animals across the world were definitely a little bit lacking, right? They tended to be a little bit long-winded and not very standardized. These are some examples from the 1500s, and this is mostly coming from probably sailors that were off around the world. Sometimes they had naturalists with them, sometimes not, and they would describe these things, or they were secondhand. The octopus is actually relatively accurate, a little bit of an elongated eye, but not too bad. The rhinoceros is a little bit less. It's missing a horn. There's a few added features and some hair in places that it's not really supposed to be. The walrus is probably the most different from the reality of what a walrus really is. But again, this was probably in an Arctic setting. You didn't have a lot of pictures, or anything. It was just sketches and just descriptions about what it might have looked like. And then, of course, scientists would try to categorize things based on these inaccuracies, and that's why early classification was a little sketchy. So now let's get into the modern system of classification, and this was shepherded in by a scientist named Carolus Linnaeus, who you might have heard of in a biology class before. He was a Swedish botanist, physician, and zoologist, and also known as the father of modern taxonomy. So let's look at what this whole taxonomic framework looks like. First of all, we start with the organism's name, and we use a technique called binomial nomenclature. It's a two-part Latin name given to each species comprising of the genus, or the group closely related to the species, followed by the species itself. And again, this was first formulated in 1736. Now, when we write these things, we actually capitalize genus and lowercase the species name, and they are all written in italics on the computer, specifically, or underlined if you're doing handwritten work. Now, down here, you'll see an example of a scientific name, Panularis argus. Panularis being the genus, argus being the species, and that is our Florida spiny lobster. Now, the way you can look at this too is just like how we have a last name and a first name. Well, in this case, the last name would be first, Panularis, and the first name, argus, would be the species name. So just another way to look at it. So now we can have this hierarchical 7-level taxa system with the most broad to the most specific. So we can actually look at this. This is, again, something you probably learned in a biology class: kingdom, phylum, class, order, family, genus, species. King Philip came over for good spaghetti is one of the classic little acronyms for that, but you can probably make up some others on your own. Kingdom, of course, is the largest group. Now on the right, you'll notice there's domains at the very bottom. We're going to get into that in a bit. But kingdoms would be, for example, the animal kingdom. This includes everything on that picture, including humans, snakes, dogs, orcas, lions, whatever, the whole shebang. That's the animal kingdom. Now, when we move up to phylum, we can go phylum Chordata. Now, if you notice there, we lose the worm because the worm is not a chordate. It doesn't have those components. So we get rid of that. When you go down to class, we have class Mammalia. Now we get rid of the snake, right? Because it's not a mammal. And then we can go to order, which would be order Carnivora. Notice that humans fall out there because we're not in order Carnivora. Then we have family Canidae, which means we've sort of gotten rid of the cat. The lion goes away. And then genus Canis. Now we're down to the dog and the wolf, and Canis lupus, which is just the dog. So this is how things sort of stack up in terms of the larger kingdom versus the genus and species. So Linnaeus grouped species into larger taxa mainly according to the visible similarities and differences that you can see with your eyes. Now, this can be a bit subjective, right? How do you decide what's important and what's not in terms of relatedness and what goes together? Look at these pictures as an example. Early on, the picture of the coral on the right, generally people thought those were plants, but it is an animal. We know that now. The sponges are the same way. You wouldn't think that's an animal until you studied it and you realize, oh yeah, it is an animal. It actually actively preys on plankton, for example. And then of course the macroalgae on the left, again, it looks like a plant, but it's actually an algae. So there were a lot of things that they were able to get right by looking at just visual, similarities, but there's also a lot that they did not get right early on due to the limitations of the time. So we have a term called homologous structures, or body parts with common features or traits. A large number of homologous structures is a strong evidence that the organisms may be related. For example, leaves in plants. There's all of these plants that you're seeing here. Pitcher plants, Venus flytrap, poinsettias, and cactus have leaves. However, they're all different in design. They've all been modified. However, they are still leaves, so they are somewhat homologous. Here over there in the right, you'll see forelimbs in frogs, birds, rabbits, and lizards. They're the same set of bones. They have been articulated and shortened and lengthened depending on the uses and adaptations of that animal, but they are all homologous structures. Now, if you've ever seen like Tree of Life with everything connected, there's some rhyme and reason to that. We use things called cladograms, which is a diagram that shows evolutionary relationships, where each clade is a grouping that includes common ancestor in all the descendants, living or extinct. Now we can also look at shared and derived characters. Now these are traits. Now if you look at the cladogram on the right, you'll see the chimp and the mouse at sort of the right end there. Now in terms of shared characters, the two lineages have in common, both of them have fur and mammary glands, right? Now, derived characters would be traits that evolve leading up to a clade, meaning the claws, nails, lungs, and jaws, which both the mouse and the chimp have, but so do other animals leading up to that clade that is the chimp and the mouse. So that's the difference between shared and derived characters. And just to point out, what we're really looking at is what happens with common ancestry. So the mouse and the chimp obviously aren't directly related, but they had a common ancestor, and somewhere along the way, that common ancestor had both fur and mammary glands. Even farther back, they had less in common. With modern classification, we have essentially moved beyond just looking at homologous structures, and now we're starting to study DNA, because of course the more similar the DNA sequence of two different species, the more recently they shared a common ancestor. So we have what we call the six kingdom system that really came into play due to examination of microscopic organisms used today. And this includes eubacteria, Archaebacteria, Protista, fungi, plant, and animal kingdoms. So those are the 6 different kingdoms that you see there. Now, above the kingdom level, we do have what we call the 3-domain system. Now, these are larger than kingdoms, and they come from molecular analysis. Essentially, we have the archaea and bacteria separated out, which the archaea includes the kingdom Archaebacteria. They're unicellular, prokaryotic, and live in extreme environments. Then we have bacteria, which include kingdom Eubacteria. This would be your unicellular, prokaryotic organisms, and they are very ecologically diverse. Now, the next domain would be eukarya, and that all organisms that have a nucleus. This includes Protista, fungi, plantae, and animal. So all 4 of those kingdoms are under the eukarya domain. So to add some depth to how we find out these relationships, let's look at embryological development. Now, scientists can look at embryological development of different organisms to infer ancestral relationships. For example, you can see here, this is a fish, salamander, turtle, and human embryo. Now, if you go to these earlier forms of the embryo, towards the left of each of the series, you'll notice that they look very similar. And then they start to differentiate as they get older and develop. Another clue can be actually found throughout the gastrulation process. Now, the gastrulation process is the process of which an embryo forms germ layers. Now, these would be your normal tissue layers that you already know if you think about it, right? The ectoderm is the covering of the surface of the outer layer. It's essentially your skin that develops that. The endoderm is your inner layers, which line your gut. And the mesoderm are the layer of cells between the ectoderm and the endoderm. This would become your musculature and other organs. Now, we have organisms that are either diploblastic or triploblastic, depending on how many tissue layers they have, or germ layers. If you only have 2 germ layers, you're considered a diploblastic organism, and if you have 3, you're triploblastic. And now an example here is the sea anemone, which is a diploblastic organism. So as that ball of cells in A starts to divide and they start to form tissue layers in B, they only form ectoderm and endoderm. And then, of course, it progresses C, D, and E until you see your modern anemone. Now here's another one, the blast pore. Now the blast pore is the first opening into the gastrulated embryo. And essentially what happens is you have that ball of cells and it sort of invaginates on itself and it creates an opening as it creates those germ layers. And that opening is either going to be a mouth or it's going to be the anus of the animal, one or the other. Now, if you are in the category of animals that are considered protostomes, the blast pore forms the mouth. And if you're a deuterostome, that blast pore forms the anus. Now, we're going to talk about the different organisms as we move forward and whether or not they're protostomes or deuterostomes. And that helps to set up clades, for example, in the phylogenetic tree. So, yay, we've named everything. Well, as much as we've been able to so far. However, how do we figure out what things are what if you don't know? And one of the ways we do that is using a dichotomous key. It's an identification tool utilizing a series of choices between alternative characters with a direction to another stage in the key until the species is identified. That's kind of a long-winded definition, I know, but essentially what it is it's a series of 2 questions, and you keep answering those questions, and it leads you along the way until you finally get to, hey, this is the animal you're looking at. So let's give it a shot using this image. Now this image, you can definitely tell it probably looks like some sort of a worm, right? But let's figure out what phylum it is found in. So if you go to the first question, it says 1A and 1B. You'll see the 2 different parts of the one question. 1A says one-celled organism exists singly or in colonies, usually microscopic. Now, does that look like that picture we have right here? No, that is totally not it. So we are going to go to 1B. Many-celled organism, mostly macroscopic, easily seen. That looks like our critter right there. And if you look over to this right, it'll say go to question number 2. So now we're going to go to the next set of 2 questions, 2A and 2B. Now 2A said the body surface with many pores, one or more large oscula or openings present, shaped varies, and generally with a rough spongy exterior. Okay, that doesn't look like us at all. Okay, now let's go to 2B. Body without numerous pores, has a mouth and a digestive tract or digestive cavity. So just looking at this image, I can tell you, yeah, it does have a mouth. It's actually on the left side of the worm, if you will. So we're going to, and it definitely, so we're definitely going to go with 2B here. And that tells me to go to question number 3. So question number 3, A and B. Question number 3A, body cylindrical and umbrella-shaped, radial symmetry, mouth surrounded by tentacles, bearing pneumatocysts or stinging cells. That is not the case here. They're antennae, but they are not tentacles. So we are going to go to 3B. Body not as described, so we're going to go to question number 4. Now, question number 4, A and B, we have a flattened dorsoventrally body and no segmentation. We definitely have segmentation in ours, and it's not flattened, it's more round. So we're going to go to 4B, body not as described. So we're going to go to question number 5A and B. Now, 5A, body veriform worm-like and thread-like, no segmentation. We have segmentation. So we're going to skip 5A and we're going to go to 5B, and it's going to tell us to go to number 6. So here's question 6. Body veriform segmented appendages when present, not leg-like. So that looks like us because we have segmentation and we have those appendages, which are actually what we call parapodia. We'll learn that later, that come out of the worm. So this would be phylum Annelida. And now we've found that this is a worm that's in phylum Annelida. All right, that is it for this note set. Remember, the main idea here, the Linnaeus taxonomic system allows scientists to use a standardized system to identify and group living organisms and to study evolutionary connections. And of course, all of those important learning goals are under there. So until next time, keep learning.